Well, welcome everyone. Good afternoon. It's great to see so many people here with us in person and online on such a beautiful afternoon. That really speaks highly of everyone's interest in this topic. So wonderful to see you all. And my name is Jerry Bowers, and I'm the VP of External Affairs and Chief of Staff here at MDI BL. And we are excited to have a conversation with Dr. Haller, Hermann Haller, who is the president of the laboratory, about a topic that many of you may have heard us talk a little bit about before, and that is art and science. And it's something that we are particularly passionate about here at the MDI Biological Lab. And we wanted to chat this evening just to sort of lay the groundwork for you in terms of, you know, I always say to folks that pretty much everything we do here at MDI BL is an experiment of sorts. And so the art and science program has really evolved over the last few years as we've kind of, you know, tried new things and tested new ways of engaging our audiences in this conversation. And so we hope that this will be really a participatory conversation where you can weigh in with your thoughts and ideas about how our art meets science program is evolving and where you see you know, maybe the potential benefits or new ideas that you might share with us. So Dr. Haller, as many of you know, Dr. Haller is a trained art historian, and so he is very passionate about connecting these two, what some people may think of as sort of seemingly very different fields, and yet we see so many opportunities for overlap and conversation, and really just a wonderful way of communicating the science that's happening here at the MDI Bio Lab. So, Dr. Haller, we have talked a little bit about art meets science, and as I mentioned, you know, it has gone through a number of iterations since we started, what, 4 years ago? I guess?
4 years ago, something like this. Our discussions, I don't know, but around 4 years ago.
So,
Roy, can we have the first slide here?
Maybe you could start, and I know you're waiting for your slide, but maybe you could start just by helping the audience appreciate why you thought this was an endeavor that was worth doing.
Yeah. I'll do this in a moment, but what we'll do is, this is the program as it stands. So I'll walk you through the program, we'll walk you through the program and discuss this. So lectures and seminars, more than 15 public lectures over the last 3 years, exhibitions, and we'll talk about exhibitions 24 and 25. We'll talk about our artists in residence. We had this year, we'll talk about museums tours, not so much about education programs, but because we are planning this. And then I'll take you on a museums tour, which is lacking here, which is Boston Museum of Fine Arts, and we'll do one glimpse of one Rembrandt painting in Boston. So now you were asking the question, why? And here, if it moves, I think, Roy, you have to help us here. Or, oh, no, I know what's missing. I should turn this on.
I think it's on automatically.
Okay. So, Jared, that's okay with you. I'll just stand and you can sit there. And then, so it's about the integration of research at MDI MDI-BL with arts and humanities. And I think you're all aware of this. Despite all my enthusiasm and despite all what you'll hear, we are a research lab. And we are very important. We are doing basic research. And now and then we do translational research because we are interested in diseases. But we also would like to integrate this. And these are some of the leading minds who have given us an idea. 1937, Albert Einstein wrote, "All religions, arts, and sciences are branches of the same tree. All these aspirations are directed toward ennobling man's life, lifting it from the sphere of mere physical existence and leading the individual towards freedom." And you can see this is old. This sentence we have stolen. Branches of the same tree is used by the National Academy of Science for their whole educational program combining humanities and art. But you can see that as an art historian, to develop a complete mind, study the science of art, study the art of science, learn how to see, realize that everything connects to everything else. That's Leonardo. I don't totally agree because not everything is connected to everything else, because otherwise we can't do focused research and develop hypotheses. But the first part is really learn how to see, and for a complete mind, you have to have both arts and science. To be honest, I didn't steal that from Leonardo, but Terry found it. It is what you have used before. So our art and science program is centered on the belief that both art and science offer legitimate ways to better comprehend our work. And discerning the differences and similarities between the artistic process. Well, that's now different. Now we're talking about artists and scientists, and this will also be a topic. And the scientific method helps refine our research strategies and better communicate our work. So it has also something to do with ourselves. When you stand in the lab and you pipette and you do whatever researchers do when they're in the lab, there is not very often that you reflect about what you're actually doing. You reflect about your work, but the process of science and the process of art is not that much different. So I told you art and science, and we have used this last year in our exhibition. This is obvious. Is this art? Who thinks this is art? Who thinks this is science? Well, most of you can't make up their mind, which is okay, because this is really fascinating. And here, I think everybody agrees this is somehow not science, but art. But I can tell you it's very definitely science as well. And this is art, but it's also at least medicine, anatomy. And this, we have talked about this when we were in Philadelphia at the museum, is definitely both art and science. So let's first talk about lectures and seminars. And this is just to whet your appetite, because these are the lecture series which we have, and some of these lectures have already been given. So Alone in a Field, you all know which painting of Andrew Wyeth we are talking about here. And what has this to do with science or medicine? Most of you know, but we can talk about this more in detail. But it's Rembrandt, and you'll hear a little bit about Rembrandt later. Albrecht Dürer, we have done Paul Klee and marine biology. That's a project where we actually want to have an exhibition in the future on. Max Beckmann, and you can go through a whole list. The knees of Michelangelo. Have you ever thought about the knees of Michelangelo and what this has to do with science? I can tell you a lot, because he had obviously arthrosis or something else with the knees. We have Mona Lisa as a patient, and some of you have heard about that, and others. So these lectures are supposedly interesting, and they combine art and science and make you think also about the history of art and science. Because I consider it, and this is also what the program is all about, that you have to have a historical understanding what you're doing, both in order to win the Nobel Prize. You know, if you don't have a historical understanding about your own importance as a scientist, it will just be by accident that you win the Nobel Prize, which most often happens. But also to be aware where we come from. And you have all heard this famous sentence that we are all standing on the shoulders of giants. And we do that. I mean, Frank Epstein was not that much taller than me, but I'm still standing on his shoulders so you can see further. But it's important that you realize where you come from, where we come from, in order to make the science better and to make us better understand in which direction we are moving. And it's always humbling also for us to understand that every generation going back 1,000 years and further had the firm belief that they were on the pinnacle of everything. I like this quotation very much. 1503, a young man in Germany, a doctor, a medical doctor, how wonderful to live in these times when we have finally understood everything. We can do everything. We know what's happening. Wonderful to live in these enlightened times. And when we think 1503, give me a break, you know, I mean, somehow this was mud and slime and there was not a lot happening. So this is a lecture series about our exhibition. So we started in 2024, very ambitious, lots of work. And you have seen this before. This was Arts Meet Science, branches of the same tree, and we've talked about that. And here you see that there are different aspects of that. When you come to an exhibition, what you expect are wonderful, impressive images. Otherwise, why should you come to an exhibition? So yes, this is impressive, both of these, but that's only one level. The other level is always educational. We all know that's a little bit boring, but nonetheless, when we try to make the world understand, then we break this down into different chapters, and we have done this year, Images of Science, Making the Secrets of Nature Visible, and the theme was, at what level do you actually see and recognize science? And we started like this. And this was curated by Emily, who just entered the room. And without her, the exhibition wouldn't be here. And lots of thoughts went into not only the exhibits, but also in the frame work, etc. So the first part, well, no, it was too far. The first part is observation with the naked eye. You can still be a scientist without any tools, just looking at the world. And we have used two examples. One example is from our axolotl people. So this is the axolotl under normal conditions, when they are in water, underwater. This is how they spend their life. How long do they live, Brian? Like this? So no need. But when the water goes away, they change into this. Now that's fascinating. To be honest, even if I wouldn't be a biomedical researcher, I would find this terribly interesting. What's happening here? You touch them, they touch differently, the skin is different, the whole animal is different. And some of you may know that this is what Homer Smith wrote all his books about. When animals move out of the water and they go on land, they change completely. And Parag, we will not talk about this, but at the moment, this is a very fascinating research besides regeneration in the axolotl. So this is science with the naked eye. And here you see another example, this fish and this fish. You can see the difference immediately. This is an old fish and this is a young fish. So we use this as aging model. You can see, do you lose all the color? I mean, your hairs are just, and this is what they look like, a little bit reminiscence of what it was to be young, but the color is very different. They're a little bit bent, so they have osteoporosis. We know that they have a loss of brain function. And so this is also with the naked eye. But then the next level is this big invention that you open up the animals. So this is from 1904. These are 3 examples from our archives. This is the shark opened up. This is a seagull. And here you see a shark opened up again, 1904. And this is when you cut your objects open. When did that start? There are all these raging discussions. Were they allowed in the Middle Ages to open up bodies and to look into bodies? They were. We think they were. It was not because they went then into graveyards and to study humans, but to open up the body to understand what's beneath that. And most of you know about Leonardo and all his wonderful artwork where he dissected the body and looked at this in more detail. And here we have this wonderful example, and we have Ms. Ships with us. This is artwork from Evelyn Koch, and this is from Harvard in 1956, earlier than that, 1948. Yeah. And here you see Evelyn Koch. And this is in the operation theater, and the surgeons are just opening the patient, and she's making these sketches, and then she transforms it into this artwork, which is both educational and a better understanding of operational technique. So I think that's a wonderful example of art and science. She actually is Maine. She went with her husband to Presque Isle, and for 40 years, they were the heart and the soul of the academic scene in Presque Isle. And in the summer, she painted in Stonington. And there's still the gallery in Stonington where you can go and admire her artwork. And then the microscope. I did this on purpose. So this is relatively easy microscopy, where we look at the skin of these 2 animals. And you immediately can see that these skins are very different. These are the whitish animals, and these are the metamorphic animals. And so using the microscope happened around 1600. And it was fascinating. It introduced tools which changed the world, not only because we could look into tissue, but the whole way we displayed this was different. We had to use numbers for the first time, because when you made a drawing from under the microscope, you have to tell people what the size of the image is. Before that, it was more easy. And now we can use this. And these are just examples from the exhibition we have up there. This is now very advanced video microscopy, where we can see this part of the body, a tubule, moving into the other part, and we can understand this. And these are other wonderful examples from Romain Madeleine's lab, microscopy. In order to appreciate this, it's not only necessary to look at these wonderful images, but you would like to understand them, because then they become more beautiful. And this interrelationship between that, we produce wonderful pictures and try to understand nature at the same time. And then last not least, we presented this, and we called it Into the Unseeable Molecules and Genes. And this is what this exhibition is all about. It looks like artwork. If this would hang in the Museum of Modern Art, it would hang on the first floor or the second floor. It would not hang on the third floor, because this is 1930s and less and lower, but it would hang on the second floor. Oh, sorry, on this. It would hang 1950s, would be something like Hans Arp, would be minimalist art, but what it actually is, it's single cell sequencing, and this is one tissue. We label each gene in each cell of this tissue, and then we dissolve the organ. We measure all the genes in the individual cells, and each of these dots represents 25,000 genes. Now that's fascinating by itself, but then we tell the computer, artificial intelligence, if you want to, could you please make comparisons between different gene sets and give each gene set a color? And when they are the same color. And then we have also distance here. So when they are really very different, put them into different parts of the image. So all of a sudden we have something which looks like modern art, but is highly complex. And Bryak was one of the people developing this in his animal for the first time, and now we're using it all the time. Talk a little bit about money. How much is each of these runs? Yeah. So you can imagine this is also very expensive. So next is, after the experiments, our artists in residence. So some of you have seen this. This is in MCBI on the ground floor, where we have this piece of art, which is from a former period. And this is our artist in residence. Michael Takeo Magruder. He's from the UK, and he's using artificial intelligence, and he is building his art on the use of these novel tools in not only science, but also in libraries and in other institutions. And this is how we recruited him. We got to know him because he's working with a gallery here nearby. And then he came here, he talked to our scientists, and he was looking, and he still is, at our science, and then transform it into the tools he is using to make the world better understood. So he was here last week, and we presented for the first time his work. He's still working on that. And you can see here examples from his previous work. So he has worked for the National Gallery in the UK for a year, and he worked about how they digitalize books. And he was also working with the British Museum, and here you see other of his expositions. And we are looking now forward to have the same here at MDI Bio. We don't have the same rooms, but we'll do our best with the rooms we have available for that. And it will be fascinating to look at that, and will give us insights from outside about our research. And then last or least, the museums tours. So we have started in museums in Philadelphia, at the Metropolitan, the National Gallery, and here you can see our small groups. It's not that easy to get into these museums. You know, you have to ask permission, and then you're limited with numbers of people, and some museums don't allow this at all. And these are us in the National Gallery. Here we are in Philadelphia, and this is the Metropolitan. This is our second visit. Do you know this painting? That's a scientist, obviously. You can see this here. This is Dr. Lavoisier. And so we're standing in front of this painting and discussing science in 1780. You know, he was the one who discovered oxygen. And we also discuss the material, the tools he's using, because you need seriously advanced tools, not like our microscopes, but really a lot. And then we talk also about the historical background. Unfortunately, he went to the guillotine during the French Revolution. He went to the guillotine not because he was a scientist. In fact, this almost prevented him from being executed. But he went to the guillotine because his father was involved in the tax business in France, and he had inherited this. So he went to the guillotine because he was involved in taxes, and you can understand that the revolutionaries didn't like this really. So with that, I'd like to spend a couple of minutes and give you a museum tour, as I promised, if I can do this, in Boston. Now we are in the Museum of Fine Arts. Most of you would walk by. This is a small painting. It's about this size. And it hangs in a corridor right at the end of it, and you actually have really to look for it. If you look for it, you stop, because there are not that many Rembrandts in the Museum of Fine Arts. So you think, well, let's have a look at that. But as I said, it's small. And it's from 1626, 1628, I would think 1628. And you can see Rembrandt was born in 1606 in Leiden, and this was painted while he was still in Leiden. He's about the age of our graduate students. And he was painting, he paints himself in the room where you can see here the painting is painting on. You can't see what's on it. And he stands there with this, what is it? A coat, bluish. But it has all the insignia of being a painter. And a very good painter. And I'll explain this to you in a moment. And so even on this level, he resembles our scientist. You know, all our graduate students are very self-assured. They're in one of the best research institutions on the East Coast, and they're doing very advanced projects, and they are self-assured, like Rembrandt here, because, sorry about that, he considers himself to be the best painter of his time. 24 years old. That's not bad. We have this from his own hand in writing, and we have also reports from other people who discovered him at the time. You can see this here. This is how he considers himself in a self-portrait at the same time. You can see that he has a problem with the eyes, because he's telling us it's not the eyes I need as a painter, it's the brain. And we'll come back to this in a moment, but you can see how wonderful he is using light here, and the hair, not bad. He was working there together. This is Rembrandt, and that's his friend and pal, Jan Lievens. And the two of them were, as a duo, the most prolific painters in the Netherlands. So this was in Leiden, which is not that far from Den Haag, and the government found out about them, and then one of the ministers, a famous Huygens, went there and described these two. They are better than any painter I've ever seen in Italy. So this tells you how self-assured they were. He went to London, became famous there, and he stayed. And this is how he painted himself. Now, what do we understand about art and science in such a painting? I've told you that when you look very carefully, the eyes are very dark. So it's the same meaning of, I don't need my eyes that much. He's also standing far away from the easel. What he's wearing is a lab coat. That's the traditional garb you had on when you were painting at the time. So this is coming back to our scientists. They're standing in the lab. He's standing in the lab. And he has here this stone, and you can see colors here. And this is where he makes his colors. So he's telling us in this portrait, this is about as important as me, because it's a big stone. And to make my colors, and that I know how to make my colors, and my colors are special, this is part of this portrait. And you can see immediately that he means it. Look at this wall. The bluish color, the discoloration of the wall, this is very artful. And then when you look more closely, you look here at the floor, you look at the light, and all of a sudden you realize that this small painting is really meant to be a documentation of how wonderful he is as a painter. And then you look more careful. Here you can see the eyes, what I just mentioned. Here you see the stone where he's making his paints. Here you see his tools of the trade. And as I said, the lab coat he has on. So it's a very self-assured painting as the young man, as becoming a famous painter. And he's also very arrogant because he's not covering up that he is poor. He doesn't have a lot of money. He's the best, but at the moment he's broke. And when you look here more carefully, you can see this. But at the same time, he's so arrogant that even the stains here and the mold, he paints this very delicately just to demonstrate, I can do this. I'm poor, you can see this, but I'm the best in displaying where I live and what I do. And then last not least, it's the easel. And now you have to understand a little bit about art theory. The most important part here is this white line. This line defines him as the best painter of his time. And the story behind this is that the two most famous painters in antiquity, in Greece, were fighting for who is the best. So they visited each other, and one came and painted grapes. And then he left. And when the other came back, he saw birds flying towards the grapes, and that was impressive. So he went back because they were very competitive, and he drew grapes, and then a curtain halfway across the grapes. And when his friend and competitor came back, he tried to move the curtain away. So that's even better than the birds. And then number one came back, and he only drew very famous story, and every artist at the time knew about this, only drew one line, a very exact line. And then his friend came back, looked at the line and says, I'm beat, he's the best. So this story that you draw one line and you do this excellently, this is what Rembrandt did in this small self-portrait, demonstrating that because the longer you look at that, the more extraordinary is it. He could have easily displayed what's on the painting, but he didn't. We have no idea. Is he thinking about it? Did he step back and had a second look? Is it white? Where are we at the moment? He leaves all this open. And I think this is very admirable for a man of 24 years old or 26 years old. And it tells you a lot about his situation. And as I said, it's not that far away from when you are a student in the lab. And just to finish that, this is 4 years later. He's in Amsterdam. He's now really successful, and he's painting for the Surgeons Guild. So we're not that far from Evelyn Koch at the moment. He's painting for the Surgeons Guild. This is the surgeons, the doctor, the medical doctor, Dr. Tulp, teaching these are the surgeons, and this is a lesson of anatomy. And we know about all these. So they are not unknown. There were famous surgeons in Amsterdam. Dr. Tulp was very famous, became mayor of Amsterdam. So this is Anatomy of Dr. Tulp. It's the Mauritshuis in Den Haag, a very famous painting. But you can easily see that it's about medicine and art. So what's the medical part? I mean, he was paid individually by all these men because of their portraits. So that was part of the salary. He was paid by Dr. Tulp because he is the major person in that, and he was paid by the guild for that, and it hung in the guild's hall for about 150 years, and then this burned down, and then it went to the Mauritshuis, and it's now one of the more famous paintings there. But you can see here the anatomy of Dr. Tulp, and what he's doing is preparing here the tendons of the forearm. Now, when Rembrandt died, poor but famous, he had 3 prepared forearms in his collection. So the forearm meant something for him, and this forearm is basically this forearm. Now, this is the most famous anatomy book for 250 years. Before Gray's Anatomy, this was the textbook. This is Andreas Vesalius, the most famous anatomist, and he trained Dr. Tulp. So Dr. Tulp was very clear, I'd like to have the same famous, because that's the first page of this textbook, I want to have the same preparation here because I am as famous as Dr. Vesalius. And everybody recognized this immediately. This is the book. Here you see the spectacle. In Leiden, I like this, in the museum in Leiden, they have the same theater of anatomy rebuilt. So you can see how narrow this all was, and you can stand there and can see them operating down there. And here you see this, so it's a wood print, 1543, very famous. But the forearm is also important because this is the same time, and this is another forearm, and some of you may know this, this is William Harvey, the one William Harvey who discovered that we have a circulation in the body. This was a breakthrough. It took in the universities for about 150 years. I'm still astonished that in 1792, at the University of Göttingen, which is down the road from Hanover, they were still teaching that we don't believe in William Harvey. I think that's academic arrogance, but 150 years later. But these are the experiments, and you can see that these books were available to Rembrandt. He read them, he bought them. So the forearm is not only Andreas Vesalius and the reference for Dr. Tulp, but at the same time a discussion of what science was all about. And all of a sudden, we have Rembrandt, whom we consider to be a painter in the Netherlands, being connected to research done in London at the time and being connected to research done in Italy 100 years earlier, or almost 100 years earlier. And to understand this, this is what art and science can be to make you better aware of your own situation. And I can't help it, this is 1632, so it's the same year. And this guy, Jacob de Gain, he was painted by Rembrandt, so that's a Rembrandt. He was looking through these microscopes, and these are insects and microscopy pictures he drew while looking through the microscope. So you can imagine the same town, the same city, Amsterdam, the same people painting each other, doing this dissection of the forearm, reading the books of William Harvey, and using the microscope for the first time, that this is in a way spectacular to understand how this all worked. And it's comparable to our campus where different students, different projects are, different students are working on different projects. And last not least, this is Maria Sibylla Romerian, and this closes the circle. This is Metamorphosis, as she painted it in Suriname, together with her daughter 50 years later. So in a nutshell, we started with this portrait in Boston, and this is how our museums tours normally work. So we start with the painting, and then we discuss and we learn about the painting and about the background and make these connections. And I think this is why art and science is so helpful, because to be honest, without being a scientist, you can stand for a long time in front of Rembrandt's Anatomy of Dr. Toll. You don't get it. But without being an artist and having an understanding of what the history is all about and what the background is, and if you don't know that the Rembrandt who painted Dr. Tulp's Anatomy 4 years before was a very arrogant young man standing in Leiden and already aware that he's the best of the best, I think this is helpful. So with that, I think we're done. 15 minutes for questions and hopefully for answers. Thank you very much for your attention. And I hope that you will come back for arts and science at MDI Bio.
That was fantastic, Dr. Haller. So you can see why our art tours are so interesting and exciting, because you always learn something new and take away some new information. But I want to come back to this sort of why, right? Why are we doing this here at MDI-BL, and what some of the particular lessons or reasons why you find this comparison or this interaction important for faculty as they think about their work, important for our students as they think about, you know, sort of where they're headed in their careers and how they might utilize some of these lessons that we're talking about to communicate the work that they're doing.
Yeah, it's a very difficult question. I made it look easy, you know, that this is what you do when you're a young scientist. But this is not how we train our young scientists. I mean, what we train is that they concentrate on their work, that they concentrate on the experiments they're doing. We try them to understand the tools of the trade. You have seen this complicated, wonderful modern art. So this is what the focus is all about. And this is what takes 24 hours of their time. But as I said before, it has to do with our history here, and it has to do with that it's useful education-wise to look at that. Let's first talk about our history. You know, the wonderful thing about MDI-BL is that it's living history. When you go to another research institution, sometimes you look at a campus, like at Harvard, and that's an old campus, and then you have the new buildings, and only the libraries are really inside the old building. Here it's different. Here we have the labs sometimes within our old buildings, and so we are living history. And when you then look at our library, which is really very old-fashioned to look at libraries, but nonetheless, you all of a sudden realize that within our walls, so to speak, there has research going on, which is now from the last century and even before, but it's still valid, it's still relevant. How often do you think read our students literature, scientific literature, from before 1990? Prior, how often?
Really?
Well, because you are a developmental biologist and you read these old papers, but a lot of the papers are forgotten. 1975, this is really, Patricio, excuse me, but this is hard to keep up with the literature of 19th century. We know that, but I think it's our duty also to communicate this and to put this into historical context. We are thinking about Dr. Proctor at the moment. I mean, 1925, first assessment of plants and insects on the island. This is something which we have to be aware of, and we have to make our students aware of. It's educational. It doesn't come naturally. When you are 26, you want to go in this direction and not to look backwards. But I think that's part of our tradition, which we have to communicate. And we do this. We do this when we have talks, and we do this when we have exhibitions like what we do, and in our lectures. So I think that's part of the tradition. Mm-hmm. Now, the second part is, does it really make you a better scientist? Well, that's a difficult question. I think so. I think that when you have a perspective of what you're doing, when you reflect on other animal models which have been used before, when you think about where science actually comes from, what was the basis for that, and you can see a living example right outside our doors now. We have a renal course from the American Society of Nephrology, and the American Society of Nephrology pays us and the faculty to teach young doctors about where kidney disease actually comes from. And the week before, we had young doctors from Harvard, and they were internal medicine, and they learned about physiology in order to understand that when they do rounds with students and they explain where we come from, that they understand the history of that. And so I think you become a better scientist if you learn about this. And what better place than MDI Bio, where it comes naturally.
Mm. Well, we certainly have seen a lot of information on the importance of sort of creativity and stimulating, this kind of thinking as you think about science. Jim, did you have a question?
Yeah, as you've been talking about, I've been thinking about a well-known dermatologist at Yale who would take his students to the art gallery to teach them observation, to teach them to see the detail. Art is educating the scientists.
Yeah. Thank you very much for that. I didn't mention that, but I'm very grateful to Yale, and then Harvard picked this up. But the Yale course, going to the museum, was basically how I started in Hanover, bringing students into not the museum immediately, but we looked at slides and we discussed painting, because that's the example. And and we could go on more about that. The real art historians in medicine are pathologists, because they stare through the microscope. At least they did this in the old times. And then they see something and they describe it in their own words. And when they're very good, like art historians, you understand what they're talking about. And if they are not very good, you don't understand anything. But they have to describe things. And dermatology is an example. You all know Sherlock Holmes, how you make diagnosis. I mean, you look at a patient, and if you're really fast, you can make a diagnosis like that. And most of the time, he's correct, at least in the books. But dermatology, you have to look at that. When you have something here and you describe it, so a lot of medicine is the art of describing, and not that far away from, and you can use paintings and everything. And the longer you look, and this is what I've done in this one painting by Rembrandt, the longer you look, the more you see, and the better trained are your eyes and your brain for science.
That's great. We did a illustration workshop this summer with our students, when taught by a beautiful science illustrator, and she made the point that you never really know your subject until you try to draw it. Like, that gives you this intimacy with the subject that you might not otherwise ever have. And I thought that was a really interesting point, sort of, to your point about really being able to see and understand, and how important.
And we have, you remember the example of Evelyn Koch. What she did at Harvard is not that they could have photographed that, but this doesn't give you the same clarity and the same understanding of the site when you do these sketches and then you do the drawings, and some of you may remember the reflections there. This is something which you would lose when you take a photography of the same situs.
Other questions or comments? I did see one online, Sharon. I think I can just paraphrase it if that's easier. You got it?
Yeah.
Oh, there you go.
So this is a question comment. Why call Rembrandt arrogant versus confident? He had genius. Arrogant means he exaggerated his skill. Did he?
This has to do with me being German, and the use of adjectives is not as exact as it should be. Thank you very much.
Other questions or comments from others? Just thoughts about, connections and between art and science, or questions specifically about Dr. Haller's talk? Yeah. For those that are interested, we mentioned our exhibit that is currently on display in the Maine Center for Biomedical Innovation. If you haven't had a chance to see that and you're interested, every Friday afternoon, starting around 4 o'clock, it's open. If you'd love to come and see it, we would be delighted to share it with you. And it features some of the work that Dr. Haller mentioned by Evelyn Kulk, who was just an amazing scientific illustrator as well as artist. Just beautiful work that she did. She was in the surgical suite at Harvard and documenting these surgeries that were happening. So maybe not something that you want to hang in your dining room, but nonetheless, a very important, piece and a way of documenting this medical progress.
I've forgotten 2 other exhibits we have there, and they are from Kobe. So, Kobe has a wonderful museum, and we are collaborating, and I've been teaching at Kobe and given lectures there as well. And we had podiums discussion on art and science because they're interested in that as well. And we have also in our exhibition 2 woodcuts, colored woodcuts from an assistant professor from Kobe on anatomy. So this is now really art and science. She is depicting what Jim just mentioned, that there are kidneys, or at least there are organs there. And we had discussions whether this is bowel or kidney or heart, or is it now aorta. And she's very open about that. She does this type of artwork because she has an autoimmune disease. So it's her way of dealing with that, both on an artistic, but also on a personal level.
Beautiful. Well, thank you all so much. I want to mention that this is actually our final in-person Science Cafe of the summer season. So we'll now revert to our virtual programming for the fall. So our next cafe will be on September 9th, I believe. And that's an interesting conversation with a former student at MDI Bio who will be talking about equity in science and governance. And she's a wonderful scientist herself and been very successful. And so we'll send out an announcement about that. It's on our website as well. If you're interested, I think it'll be a great conversation. We're really excited about that. But Dr. Haller, thank you so much for sharing your enthusiasm for art with us today.
Thank you.